This experiment uses flow cytometry to detect and quantify nuclear proteins in isolated and immunolabeled nuclei from neutrophils. To purify the neutrophils from human peripheral blood, remove the red blood cells with dextrin, followed by density gradient centrifugation of the plasma. Then, using anti-receptor antibodies, stimulate the purified neutrophils via integrins or FC receptors. Proceed to isolate the nuclei and fix the samples for immunolabeling with specific antibodies against the nuclear factor of interest, for example, NF-kappa B. Finally, analyze the nuclei by flow cytometry in order to detect small changes in nuclear factor activation. Results are obtained that show stimulation of neutrophils via their integrins leads to an increase in nuclear concentration of the transcription factor NF-kappa B.
Signaling pathways that lead to nuclear factor activation are usually studied by reporter gene assays or by electrophoretic mobility shift assays. In primary cells, often these methods are not suitable. Flow cytometry offers rapid detection and quantification of nuclear proteins in isolated nuclei.
Start with 10 milliliters of freshly collected blood from healthy adult volunteers. Pipette 2 milliliters of 6% dextrin T500 solution into a 15 milliliter conical centrifuge tube. Then add 10 milliliters of blood. Mix by inverting the tube 2 or 3 times and allow 45 minutes for erythrocyte sedimentation by gravity. In a fresh 15 milliliter conical centrifuge tube, place 5 milliliters of Ficol Pac. Harvest the leukocyte-rich plasma that formed above sedimented erythrocytes. Carefully layer it on top of the Ficol.